Hi, this is Dr. Ben Morrill. Welcome to episode 65 of Reptile Genetics Weekly. We've got some really interesting stuff to talk about today, some cool stuff. Got a guest here to talk a little bit about some strange stuff with puzzle and clown. So I know some of you have struggled with producing a puzzle clown, and we've got some more. I don't know that we'll really answer any questions. We might just make more questions, but excited to talk about that. Also, we have an exciting announcement. We do have 2 more tests that are available. So yeah, lots of good stuff to talk about today. As far as updates for results, fast tests are going out pretty much every day. The panel results should have all gone up. So if you had we were expecting panel results and you haven't seen them yet, let us know. But the next round of panel results will probably come sometime next week. So I would say like the Friday of next week. So about a week and a half from now, we'll have the next roundup. So, but yeah, that's pretty much it for results. Kayla, how are you doing?
Hey Ben, I'm doing good. Almost forgot how to unmute myself for a second. I have a quick key for it. I just forgot for a moment. But anyway, I'm doing well. I actually sold my very first hatchling ever this weekend. It was actually somebody you sent my way, but there's a picture
Yes.
of my little het lightning boy with his pretty mama chickadee. And Jay and I were like, when they were on their way out, because they came to the apartment to pick them up. And we were both just like, look, our first grandkid is leaving the house. So
Yeah,
so
they grow so fast.
they do. Now I just gotta wait for I have one other that's gonna be going this week that's will possibly go this weekend. And then the other 2, well, they're gonna make me work for, making sure they get established. So there's always 1 or 2 in every clutch, I swear. And so yeah, that's how it is for me. But we have well, actually, let's get to our business stuff before we get to our guests. So Clutch and Morph Market both have some really nice bundle tests and discounts that you can get on those. I don't know if you wanted to say anything more about
Just
those, Ben.
but any time you have animals where you want to test for multiple mutations, if you're trying to make triple hets or quad hets or even more complicated than that, thankfully the pick 4 we did a couple of months back was to kind of pave the way, see how helpful this was, and a lot of you use that. And so both Clutch and Morph Market came up with ways for you to be able to order in those bundles. So you can customize, pick your own 3 or 4, whatever it is that's going to be helpful to you, and get a good deal on that. So
That's awesome. And let's see, we've got, of course, bulk ordering. This is always a thing. If you're ordering 25 or more shed tests, give us an email. We might be able to work out an even steeper discount for you. So order in advance if you've got some clutches coming, or just got some. You can use them now or later on down the line.
I was
Go
just going
ahead.
to say, and Sean wanted me to make sure to mention, he's the one that does these deals for people, the bulk ordering deals. He said that make sure people understand, if you know exactly what it is you want done. If you have 20 you want to run, but you're doing 2 tests on one, 5 on another, 3 on another. If you're able to email that and say specifically what tests you want, then not only will you get that quantity discount, but if he knows, like if you're doing 10 of them, you're running for 3 genes, he'll give you that 3 gene price, and then he'll take 1% off of that. So as
Nice.
much planning ahead you can do, and let him know specifically what tests you want done on each sample, that's a better deal he'll be able to give you.
Yeah, that's great. And of course, we do have 2 new tests that we're proud to announce, Barnhart Black Pastel and Pastel. So Ben, do you want to talk about those?
Yeah, very excited to have that. So there's a lot of people, and we had, of course, Tom Barnhart on here to talk
Yep.
about the Barnhart line, and we started working on this, and thankfully this was one of the easy ones that popped out clean and works well. And so now we do have a way for people to be able to tell if they do indeed have a Barnhart Black Pastel. And then the Pastel, I think, is helpful for people that maybe more so want to remove Pastel from their breeding colony, but either way, sometimes you're not sure if you have a super Pastel or a Pastel, and now we've got a way to test for that.
Yeah. Plus, I know that like that makes it easier for you to produce exactly what you want rather than overproducing things that you don't. So that
Yeah.
works really nice.
And those two in acid aren't part of the big panel yet. The big panel is still that 30.
Mhm.
But over the next couple of months, we'll try to get it where acid, Barnhart, black pastel, and pastel will be there, and hopefully we'll have some other new ones here pretty quick too that I'm working on.
Oh, cool. All right. Well, it's time to introduce our guest today, and that is none other than Mr. Justin Kobilka of KINOVA Reptiles.
Hey guys, happy to be here.
Yes, thank
Good
you
to see you.
for coming on. Today, we are going to talk a little bit about a clutch that Justin had that kind of gave him some odd results, some unexpected results. And so go ahead. Maybe the best way, Kayla, is to, or maybe even before that, Justin, if you want to talk a little bit about why you chose this pairing.
Yeah, let me just introduce it a little bit. If you've been following my Patreon, we've been talking about this clutch a lot because it's really surprising for us. It was a pairing from if you want to bring up the mom photo, what is it? Inchy red stripe. No, sorry. Let me get this right. Inchy fire DG cryptic. So it is
Ooh.
a double recessive for homozygous cryptic, homozygous desert ghost, plus fire and Inchy. I bought this girl a few years back, and she's just been amazing, and she's gorgeous, as you can see there.
Yeah, she's pretty.
Yeah, I loved it. And I was really wanting to get some more double head DG puzzles this year. So I bred her to my red stripe puzzle. And you can see the pairing photo here. And he's gorgeous as well. I got him from a local friend here, a customer, and just a great pairing. We hope to make some double heads, or triple heads actually, with the cryptic DG and puzzle. And when we went to, expected nothing more than that, but when we went and cut this clutch, we did it on camera for our Patreon. The babies looked unique. So, and they basically look like cryptics or kryptons or, lesser versions of puzzle, essentially. We're all in that same kind of ecosystem.
Yeah.
And it was very surprising. It's not a Partho clutch or anything like that. We had all the genes from the male with the red stripe. This is an Inchy red stripe triple het, but it looks like a visual cryptic, and it is very unique and very interesting. All the babies of the clutch had some sort of visual cryptic characteristics. This is the fire. No, sorry. This is what is this one exactly? Yellow belly. I don't have the IDs in front of me.
Oh, sorry. I clipped him out so I could zoom in the
It's okay.
animals.
That's okay. In the end, the incomplete dominants don't matter. The point is that they all look like Krypton type animals.
They do.
There's no explanation for that from being triple heads. And here's another great version of it.
Yeah.
So in the end, we basically were like, what's going on here? We sent these to Ben and Sean and said, can you make sure? The main thing we wanted to make sure was that they were triple head, of course, that no Partho kind of situation was going on. And second of all, we wanted to be sure that the dad, the red stripe puzzle, wasn't somehow het clown, which would have made them into visual kryptons potentially. And first of all, we checked them for the triple head. They came back as het for cryptic, DG, and puzzle. And then we tested them for het clown. They're all negative, and dad was negative for het clown. So there's no krypton chance here. That's what I can visually see. Now, Ben is the expert on what possibly could be going on, why we're getting these weird results when we're mixing cryptic and puzzle.
Yeah. So are those the next slides, the things that you sent to me, Ben?
Yeah.
Yeah.
Don't bring them up quite yet, but yeah. So, there are a few different things that can happen, and we've kind of learned as we've done more genetic testing, and the group from Canada and Europe, and the various things that we've been doing. We can see, like an example with axanthic, there's at least three different genes on three different chromosomes. When they're mutated, you can make axanthics. So there's definitely different things that can go on different places in the genome that can give you the same look. Well, in this case, there's kind of two main things that I think could be happening, and we're definitely speculating right now. We'll have to do more breeding and more testing, but one of them, if you want to bring up that first picture, Kayla,
Yeah.
one is called one possible explanation is epistasis, and what this is a specific interaction between two different genes. And if you want to go to the next one, Kayla, a really good example of this is with Labrador retrievers. And so there's actually two different genes that play a part in whether you get a black lab, a chocolate lab, or a yellow lab. So one is important for the development of the pigment, and then the other kind of controls how well that pigment is actually made. And so it's like two different things that have to line up. So if the animal is homozygous recessive for that little e, then you get a yellow lab. But if it's heterozygous or homozygous for the dominant e, then you'll either get chocolate labs or black labs. And then the difference between the chocolate labs and black labs is the other gene, that B allele, I guess, B alleles. So you can have where two different genes in two different locations can affect the look of something. And
interesting.
There's definitely, I've been doing more reading, studying on pigmentation genes and pathways and things like that. And there are definitely pathways where there will be, 5 or 6 genes that all work together to make like the melanin, how much will be there or where it will go to or whatever. And so it could be that puzzle and clown both do something, puzzle, clown / cryptic, I guess, but they do something, those two genes do something in that same pathway. And so even though they look like, as far as we can tell, they're different genes on different chromosomes, both what we can see and what other researchers have seen. It could be that they play a part in the same pathway. And so if you're heterozygous for two things in the pathway, it could actually cause a change in what you're seeing in the animal. Kind of like what we talked about a few episodes back. We can tell you what the DNA code is with genetic testing, but we can't tell you what it's going to look like at the end of the day until breeders do the pairing. So this is a really cool, interesting breeding that Justin did, and definitely surprising. And one question I have for you, Justin, is if you had done this pairing, 5 years ago and there was no testing, how would that be a difference, to what you're going to do with these animals now?
5 years ago, I don't think we knew what a krypton was. But if we did it 3 years ago, I would have assumed that either the mom, which I thought was visual cryptic, I assumed either she was a krypton, or I would have assumed that the puzzle male I used must have been het clown. And it would have taken a couple years of trial and error to discover that both of those were incorrect conclusions.
Yeah, that's definitely surprising.
Yeah.
So what kind of thoughts and comments did you get from your Patreon members? I'm sure there's some people.
Curiosity. And what everybody wants to know is, everybody's so curious about the puzzle clown project. And does this mean anything specific for that project? Like, are we still at the same place where we're still trying to, make them and figure them out? Or does this give us any insight into that combo as well? Or is it separate? So that'd be the thing everyone wants to know.
Yeah. So, if it's epistasis, then, it really shouldn't, there should not be any increase in difficulty in making the double homozygous animal. So I would guess that that's what we're going to see, is people have just been unlucky, or maybe people have made them and not gotten them tested. I don't know how likely that is, but maybe they produced something and it didn't look like what they thought a cryptic or a puzzle cryptic or a puzzle clown would look like. So they, didn't have it tested. That would be something I would be very interested in, and we'd happily run some for free for people that have, puzzle clown or puzzle cryptic clutch, where they could have potentially produced the double recessive, that double homozygous animal. That's a possibility, because if those two genes in different places are causing weird interactions, it could be that double homozygous animal doesn't look anything like what we thought it would. It could also be kind of like a spider blackhead thing, where they both have very different looks, and then you breed them together and that spider blackhead just looks like a normal. It's possible. So I would like to run some from clutches from people that, even if they don't think that it really is the double homozygous, it would be nice to run. And if we just see, no matter how many pairings we get, you just keep getting, hets, double hets. We don't actually get the double recessive. The other possibility, which would make it difficult to get the double recessive or that double homozygous animal, would be if there was a DNA translocation. So this is another thing that's very unlikely, but the work that we've done, and I would assume, I don't know the details behind other groups that have looked at this too, but what we can see is puzzle and clown / cryptic, they're on different chromosomes. But what could have happened is when that puzzle, it could be the puzzle or it could be the clown / cryptic, that gene could have actually broken off and translocated and moved and is on the same chromosome now as puzzle. So they could actually be right close to each other, and we wouldn't necessarily be able to see that, because when we're doing all of the figuring out what's on which chromosomes, we're mostly using normal ball python DNA. And so it's not very likely that a translocation would happen and that gene would still be active and do the same thing that it would do somewhere else. But it's possible that they're near each other. And if they were near each other, if there's a translocation that puts them right by each other, then that would make it so it would be difficult to get. It would be kind of like trying to produce a female banana from a male maker male, because those two signals are right next to each other. And on one chromosome, you'd have het for clown or het for cryptic and normal for puzzle. And then on the other chromosome, you'd have the het puzzle and you'd be normal for clown cryptic, and you would never be able to get them both on the same chromosome, which would mean, it would be harder to get that double homozygous. So that's possible, but not.
Let's hope that's not the case.
Yeah, if that's
hopefully
the case,
not.
It'll be very difficult. But the good thing is, if that did happen, which, like I said, is a low probability, but if that did happen and someone eventually does get it, like we can get a female from a male maker, then that animal, all of its babies would have both signals. So if you bred that double homozygous animal, the babies from it would be either double hets or normals. So they would get both signals or none. So, well, I guess if it was double homozygous, every baby would be like that. But when those offspring that are double hets bred, then they would always pass the het for both to every baby. So it would be the ratios would be very different than what we're used to seeing because of those two things being linked. So I would say, because we've had a hard time producing it, if we do the testing and we see the potential double recessive animals from those clutches, actually none of them are double recessive, then this could be what's going on. But like I said from the beginning, I think it's more likely it's epistasis, and we'll get it at some point, either something that looks like what we expect, or we will get from testing, we'll be able to see that we got it and we just couldn't tell by looking at it.
Well, we'll keep trying, and you scientists keep doing your thing, and figure it out, hopefully.
Yes,
that'll be exciting.
Well,
and if and when we find out, we'll probably have to do a video where we kind of explain this, but in ball python terms, because I know it's probably a little it's a complex subject that you don't always wrap your mind around, especially if you're not a geneticist like Ben. So
Well, for me
hopefully
too,
we'll
because
find out.
I've been
Yeah.
mostly thinking in ball python genetics for like 20 years.
That's fair.
I haven't thought about basis very much either. So it's definitely complicates things. And if there's a translocation, that complicates things. But you do enough different mutations in a species, you're going to end up getting some weird stuff.
Right.
Yeah.
Well,
All right.
one more permutation of this. Let's go over for the audience that I've been thinking about is if this can be replicated, which hopefully it can, where het puzzle, het cryptic makes a visually different animal, then we essentially have a version of that cryptic look that is non-allelic, that could reproduce itself. So theoretically, if I was to breed this animal, and just correct me if you're a flaw in the logic, if I was to breed this animal and I was to get another one of these, I could theoretically know it's double het, and it could reproduce itself.
Yeah, that logic makes sense. It's like a bumblebee.
Yeah,
two different mutations on two different places that causes that. That'd be just like breeding a bumblebee, and 1 in 4 you get another bumblebee.
Fair enough.
More layers to this onion. Okay.
Yeah. And so we actually had this shed donor shout out set up like 2 weeks ago for Tom Harbin, but we got very tragic news about his passing. And so this shed donor shout out is much more bittersweet than our previous ones. So Ben, did you guys want to say a few words about him?
Yeah,
we love us some Tom Harbin.
Yes, I was just going to say I was just talking to him last week about 2 different orders that he had in to get results, and it's definitely that weird feeling. I'm sure many of you have had that, where there's someone you were just interacting with, and
yeah,
so yeah.
Tom's a legend in our industry for his kindness and consistency, and just his kindness. Really, kindness is the word that comes out. He's kind to everyone, and he would spend so much time talking to not just the vendors, but teaching people and taking new breeders under his wing. And I've known him for 20 years now, from the very beginning, because he was kind to me, and you never forget that. So rest in peace, Tom, and huge condolences to his family. What a loss.
Yeah, the Harbins won't be the same without him, but I know that his family will definitely continue his legacy with all of the snakes he has and he's raised, and he certainly made an impact on the hobby. I remember when I went down to Arlington last year, he just greeted me like out in the middle of a crowd, and I was like, "Oh my gosh, it's Tom!" And we chit-chatted, and just wonderful guy. And we really lost an awesome person from the hobby. So my condolences, all of our condolences to his family and loved ones. Yeah. All right. So the downer, but it's good to remember an amazing guy. So I think that's all we have for today. So everybody go give your loved ones a hug. Go love your snakes. And do you guys have anything before we call it a day?
No. Thanks for being on, Justin, and
yeah,
thanks for all of you watching.
Definitely.
All right. See ya.
All right. Well, thank you guys, and we'll see you next week. And outro in 3, 2, 1.